Greetings and thanks to our panelists and the many registered participants joining today's discussion. As director of Caltech's Resnick Sustainability Institute, it felt especially important to engage with our community near and far during this Earth Week, one which marks the 50th anniversary of the initial launch of Earth Day in 1970. This has been an incredibly exciting year on Caltech's campus with respect to bold new research and educational initiatives focused on climate change and the stewardship of natural resources, initiatives energized by a historic pledge from Linda and Stuart Resnick. I encourage you to visit the Resnick Institute's website to learn more. Shifting to the topics of the day, the climate crisis presents a global health crisis with many faces, one with consequences that will ultimately reach every member of our society. The current pandemic, devastating on many levels, painfully underscores the need for the scientific community to engage with and to impact highly intertwined health and environmental policies. This will take new science and advocacy, of course, but equally importantly, it will require prioritization from a broader public that decides who leads and the policies such leaders then feel compelled to pursue. For this first panel, I'm joined by my colleagues and friends, Pamela Bjorkman, David Baltimore, Alice Huang, and Rob Phillips. They're world-renowned for their collective work in molecular and structural biology and biochemistry, including virology. To kick things off, the panelists and I agreed that we needed to deliver a short tutorial on the inner workings of the so-called coronavirus to bring everyone up to speed quickly on the biology. We also agreed that any member of our first panel could give this tutorial exceptionally well, given their profound expertise. But to make sure that I too understood the basics of this virus, I was charged with doing the short tutorial. This will no doubt amuse some of my Caltech colleagues. Okay, so what you see on the slide that I'm showing you here is an artist's rendition of a human cell along with some virus particles. The artist in this case is yours truly. I drew this image on my iPad yesterday, so I'm to blame if things are correct and or if you just don't like the image. What you see wrapped in gray is a human cell, and so that's the big oval that you see, and inside that cell is my depiction of some of the essential biological apparatus that are going to be hijacked for virus replication. What you see in the red circle enshrouded by blue spikes is the now familiar image depicted in my own rendition of the coronavirus, AKA SARS-CoV-2. The blue spikes are proteins on the surface of the virus that interface with an orange protein in the cell membrane of this human cell, and that molecular interface causes the virus particle to lock and anchor to this so-called orange receptor on the human cell, such as a cell in the tissue of your lungs. Adjacent to that protein receptor in light blue is another protein that acts as a molecular scissors that can snip a part of this protein at a peptide bond, and that causes an interesting and rather fascinating molecular contortion to begin to occur, by which ultimately this virus particle can enter the inside of the human cell. And so that entry is its line of attack. Once that virus particle is inside the cell, you can see that because of this acrobatics that it had to go through, it is now wrapped by the same type of material that the outer sphere of the human cell is wrapped by, again shown in this gray sphere. I've cut a slice through the virus particle so that you can see a depiction of what's inside it, and that's a strand in black of viral RNA. And so that viral RNA is its genetic material that is required for this virus particle to reproduce. And it then hijacks the human cellular machinery to do that. As this particle sits inside of the cell for a period of time, it begins to cure, so to speak, and begins to open up like an egg. And as that egg opens up, out is spit this viral RNA. And that viral RNA is the critical element for reproduction. That piece of viral RNA, once it's in the cytoplasm of the cell, can encounter two types of machines, one shown in light green and one shown in purple. The machine in light green is one that translates the viral RNA. So it reads the viral RNA and tells things to start making protein, the type of protein that the viral particle needs to reassemble and then infect other cells. Once those viral proteins are made, they are then snipped by another pair of enzyme scissors into the constituent peptide pieces that can ultimately be assembled in another compartment of the cell, and that's shown here, so that you now begin to have the protein constituents that you need for a virus particle. But that's not enough for a virus to go on and do its business. You also need to reproduce the viral RNA, and that's done at this machine shown in purple, which is essentially a Xerox copying machine. The viral RNA is copied by this machine to make new copies of viral RNA, and then those find their way to the same place in the cell where the protein molecules have been assembled. Once that happens, a reverse of the molecular acrobatics occurs and the virus particle exits the cell, and this can then repeat itself once, once and then twice and many times, so that you make many virus particles that can go on and infect other cells. And once those repeat, you begin to have what we call a viral infection. Now, there are a number of drugs that you're hearing about in the news, and some of them may prove effective against the coronavirus or SARS-CoV-2, despite the fact that they were developed to treat other diseases. We don't have time to go through all of these, but just as an example, there are a couple of drug molecules, one known as remdesivir and one known as favipiravir, which you can look up by Googling them. And what these drug molecules are thought to do is to interrupt viral replication. And that's shown by this lightning bolt here that stops the replication function. And that then, shown by these other lightning bolts, prohibits the reproduction of all of these virus particles, and in principle, then can prevent an infection. Clearly, the advantage of these types of drug molecules that have been pursued for other reasons is that the FDA has already explored many of these, and so some of their toxicities within humans have already been studied, but we're clearly at an early stage to know what works, how well it works, and what the side effects are. So clinical studies are rapidly and desperately needed. So that's my short tutorial on viral replication. You've also heard a lot about antibodies and vaccines, and for that, I'd like to turn things over to my far more knowledgeable colleague on this topic, Pamela Bjorkman.
Okay, thank you, Jonas. That was great. Just a very short discussion of vaccines. Basically, what you're trying to do is jumpstart your immune system to recognize something about a pathogen before it ever encounters it. So if the immune system can be tricked into thinking that it's infected, even though it's not, it will start to produce, it will start procedures that would ultimately result in antibodies. And these are one arm of your immune system. They're specific for particular pathogens or particular viral proteins. And so this is just a depiction of what might be going on if you're trying to make a vaccine. You would inject something, and that something, I guess my cursor doesn't work, but it would be on the left side of the slide. You would inject something that resembles a virus, but it is not in itself infectious. That would then trigger your own immune system to go through all kinds of things that I'm going to gloss over a lot, but resulting in many forms of immunity that include these antibodies. And some of those antibodies are then specific for the actual virus. Let's say you were trying to make an antiviral vaccine. Yes, thank you, Jonas. And so that orange antibody there might bind to that orange spike protein. If it did that, then if you remember Jonas's diagram, if you were infected with the real virus and you had an antibody, it would block the interaction with that cellular receptor that's shown there, and then the virus couldn't get in. And so the idea is to trigger your own immune system to make responses in advance of seeing the actual pathogen. So some comments about this. As you know, we have some vaccines that work very well. We do not, for example, have a vaccine against HIV. That's actually what my lab's working on, and this is a depiction of what you might inject, something that might look a little bit like HIV. And basically the reason we don't have a vaccine against HIV is not because people haven't tried, it's because the virus makes so many different mutant forms. There are so many different strains of HIV that it's really hard to imagine how you could make something that would be protective against all those strains. I think the good news here is that the coronaviruses do not make as many strains, anywhere near as many strains as HIV. And in fact, in Jonas's diagram, when he showed you the reproduction of the RNA by the purple machine right there, that is a polymerase, meaning that it polymerizes the nucleic acid bases to make more RNA. So that happens, and that's error prone, and the errors produce mutations. But the encouraging thing is that coronaviruses actually have a proofreading activity so that they correct a lot of their mistakes, meaning that they are less error prone than, say, influenza virus or HIV. So I think we could make a vaccine. The question is, how long would it take to actually test it and make sure it's safe and efficacious in people?
Great. Thanks, Pamela. Okay, with that, I'm going to unshare these slides so they're not, let's see. Right. Okay, so with that, I'm going to stop sharing slides, though we may call up slides if they're needed for the panel discussions. And I'd like to now move straight into some of the questions for the panelists and then try to get through this so the panelists can welcome some questions from the participants as well. So David, if you don't mind, I'm going to tee off with you. You and other members of this panel have extensive experience in studying HIV. How has science progressed, say, since the early days of the 1980s to better position us to find a vaccine for SARS-CoV-2? Do you care to comment on that?
Well, HIV and CoV-2 are very different viruses. Pamela discussed that a little bit. And they react to the immune system in very different ways. So we don't have a vaccine against HIV, in spite of 35 years of effort by some of the world's smartest people. And we're worried that when we try to make a vaccine against CoV-2, that we're going to find surprises. So Pamela may be right that we will make a vaccine against that virus. I certainly hope so. But we don't have one yet. And until we get a number of candidates, test them all against the virus, find the best one, build up huge stockpiles of it, spread it around, and get everybody in the world immunized, or at least some large fraction of the people in the world, we won't truly be able to say that we have a vaccine that blocks infection. We can do that for measles virus, we can do it for a polio virus, we can do it for smallpox. We would love to put COVID-19 in the same class with those diseases. The reason that we're not sure we can make a vaccine is because the virus can mutate. It can change itself. And so it could avoid the antibodies that the body makes against it. We're hoping that that's a minimal problem. And the fact that the virus does have an ability to catch its own mistakes suggests that that may be less of a problem than it would be for other viruses, particularly like HIV.
Right, so
but I think if we look at the situation we have in front of us now, that if a vaccine is, call it 18 months away, we've got to do something in the interim to begin to protect the population, to help people get over the disease, and that would be drugs. And so there's a lot of effort being put into making, in some cases, very innovative drugs to block the virus.
Agreed. And if we had time, that would be another, I mean, that is the subject of another interesting panel that would include an additional cast of characters to dive into what's involved in trying to discover and then scale up the synthesis of drug targets. Thanks. So you've commented a little bit on this already in terms of how things unravel. And I know it's hard to look ahead. One way they could at least get attenuated is through the development of promising drug molecules, or better yet, repurposing of known drug molecules whose toxicities and efficacies are known in other contexts. And so we remain hopeful for that, but the news is still sobering there. So time will tell. And frankly, the news is sobering as well as confusing if you follow the news on hydroxychloroquine. So, you know, this is another question for you, David, but I'd also encourage any of the panelists to chime in. COVID-19 dramatically reminds us, at least in my view, of the inescapable interconnectedness of the globe. I think that must be evident to everyone. The political climate, and I editorialize here a little, I admit, but the political climate in this country and elsewhere seems to have retreated from global partnership in recent years. Do our panelists anticipate cause for hope? And Alice, we may get to this even further on a later question, but do the panelists anticipate cause for hope that this global pandemic might nucleate greater global cooperation on human health, and ideally human health in the broader context of the environment and ultimately climate change? Does anyone care to comment on that? I'd also like to open the discussion for how universities might play an enhanced or stronger role via strategic partnerships and networks to facilitate this. So Dave, I'll start with you if you have any thoughts to add here, and then I'll open that up.
Getting international cooperation is really difficult. And I think at a time of crisis of the sort that we're facing now, it's even more difficult because each leader feels a responsibility for his or her own country, and often wants to blame somebody else, and that's usually the country next door. So it's not a great time to be looking for cooperative activity. But once we get this calmed down, I think that's going to be the first order of business, to try to find ways that we can all work together, because a pandemic of this sort shows us that our whole world is interconnected, that what happens in China and what happens in Singapore and what happens in France and what happens in the United States are all linked. And this isn't going to be the first, this isn't going to be the last such virus to emerge from the wilderness to attack the human population. That's something that's going to happen again and again over time. And we need to cooperatively work to prevent that from overtaking our civilization. And this should be a lesson that we never forget.
Agreed. I'd like to open that topic to any of the other panelists that might want to comment. In particular, if we want to think a little bit about Caltech and other institutions of science, universities, national labs, otherwise, both in the US and abroad, you know, we tend to collaborate by nature, and yet these really large-scale collaborations that might be needed to sort of weave things together, I'd like to explore if there's a role where we think we can do better, and just get people's thoughts on that.
I might chime in just to say that
Great.
for the last 5 or 6 weeks, actually, we've been running this coronavirus discussion group, and actually David and Pamela have been part of those. And I would say unequivocally, and I'm very curious to hear what the others have to offer, I've never seen so much interest in trying to unite in common cause. I really like the way that Newsom puts it without being political. He overuses sometimes the word meeting the moment. And many people have been rising to the occasion. And I have actually called my dad just to say how remarkable it is to be on a phone call with people that, well, I should be careful just to not cause trouble, but people from countries that normally don't hang out together. Let's just put it that way. I've seen incredibly intense commitment. And so I like that grassroots aspect. And I suppose that's sort of the counterpoint to what David said. I'm not at all trying to challenge his point, because I think it's right. But at the level of individual scientists, it's really interesting to see the commitment to working together. Incredibly interesting, impressive too.
You know, I can't help but want to also, I didn't have this question jotted down, but based on the discussion, I also just wanted to either state myself or be interested to hear anyone discuss, you know, while we seem to have trouble as a government interacting with other governments at times, especially on a global crisis, and we're not the only government suffering that, in science, our laboratories here at Caltech, and indeed laboratories all across the United States, are filled with people from all around the world. And we so desperately benefit as a society in the United States, and our campus in particular, from the hard work and brilliance of all these people that come here to work with us and share in common cause. So that always gives us hope as academics. I don't know how to translate that to common cause in policymaking, and that requires a next level of people to pick up the baton and run with it. Anybody have anything they want to add to that, or is that?
Well, you know, scientists have been known to simply work together across borders that are politically hot. I remember during the Cold War, we had scientists in the universities in the United States working closely with scientists in Russia, in spite of the fact that the governments wouldn't talk to each other. And they worked for peace. They worked for collaborative activities. And it really showed that scientists can work together even if their governments don't.
Right, and I'll simply note that a number of universities and also larger consortium have engaged Caltech, and we've committed in terms of sustainability and climate change action to participate with a number of these universities, translating that into real solutions rather than logistical bureaucracy are issues that we all face when we, you know, it sounds good, but then translating it to impact. But we are, as an institute, the Resnick Institute, and also as Caltech, pursuing these kinds of opportunities in the broader context of sustainability. And you could imagine that translating into the human health arena as well. Okay,
I can
go ahead.
imagine, Jonas, that science diplomacy is something that is extremely important, and universities have been very much involved in it because we have people from all over the world in our labs and in our classrooms. I'm always excited when I see a picture of the people working in the JPL labs. You see people from every country there. And when people open their mouths, all sorts of accents come out. And that's something that this particular pandemic has really showed us, is that we have a virus that sees no boundaries. And scientists in general do work without boundaries as well.
Thank you. I think your panel agrees with you wholeheartedly.
Yeah, I could just add that I'm in touch with some scientists in China, and they were worried about what might be going on in terms of my safety and the safety of my lab. So they sent me from China 395 masks, which I donated to hospitals, by the way. But
yeah.
So there's no problem with the scientists.
I'll also echo that. A former postdoc of mine reached out to see how he could help in similar ways. And I think many of our colleagues, from what I hear, are getting contacted that same way. It's very heartening, actually. Okay, so let me shift the conversation to modeling, and that'll shift us over, at least to start, to Rob Phillips. So Rob, scientists and the broader public alike keep hearing in the news media, more than they ever thought they might, about epidemiological models concerning COVID-19. And if you read most newspapers, or at least some of the newspapers I read, you will even find articles that allow you to interface with graphs via the wonders of the internet, where you can put in certain hypotheses into your model or certain boundary conditions, and they predict fatality rates, infection rates, etc. And then these models, or a subset of these models, keep finding their way into policy decisions. They find their way in and out of policy decisions. Can you provide a little bit more to this group of participants and to the panel, a more quantitative perspective on what's in these models, what's working, what isn't working? Are there missing pieces that are essential? And if there are, what's a pathway to getting these pieces and improving these models? And can this be done in real time here?
No, you're not being,
Sorry.
you're muted, Rob.
Yeah, I got it. So thank you. Thanks for the question, and I think it's a very interesting one, and you're absolutely right. And every day, almost in the New York Times, you see something related to a model. So just to frame the discussion, I would say that there's two categories, broadly speaking, of things that people are up to. One of them I would call empirical fits. In other words, they're basically trying to fit a mathematical function either to the cumulative cases or the fatalities or some other such measure, and to try to use it as a way to forecast what's going to happen next. They may or may not account for things like us actually sheltering at home and so on. And you probably have heard about the University of Washington empirical fit, which is based on a particular kind of math that doesn't really matter. And so the idea there is to try to make a forecast. The second class of models are ones that attempt to put in some realism about the actions of people. So you have some population that's susceptible, some populations exposed, some populations infected, some populations that's now recovered, and you can write down a mathematics of that. And so that's a kind of very different worldview where you're trying to predict things based on the laws, if you will. And so writ large, what I would say is there's very interesting analogies with two previous episodes in history. I'll try to keep it brief, but it was a very hard slog to get to the point where we could do things where Caltech can send us all an email saying, hey, it's 2012, please show up over at the athletic field because there's going to be a transit of a planet across the sun. And you go there and it happens. That was not true in the year 1750 when Euler and others were trying to figure out whether the Newtonian laws were even right. So it's a long, hard slog. The other example that I think is probably even more timely is just weather. And that also has been an extremely interesting progression, and of course is directly related to the topic at hand about sustainability and climate. So what I would say is that we have more than 100 years of people doing models. In fact, the vaccination models go all the way back to Daniel Bernoulli, believe it or not. He actually was thinking about smallpox and the role of vaccinations in suppressing infections. And so, this has been on the books for a long time. And one of the interesting features, and I'll stop, is just to say that these models have to account for something that the planets don't do. And that is that, all of us are watching the news and then we have our own behaviors. So that makes these models more tricky in some sense than the models that you would write down for Mars circling the sun, or for the moon going around the Earth and the Earth itself going around the sun. So there's a lot to digest there. And what I guess I would say as a final thought is that if we use the weather as an example, in the last, say, 50 or 60 years, we've had increasing spatial and temporal resolution in our measurements. And I'm just very hopeful that projects like the Global Virome Project are going to now be doing even more rigorous surveillance of viruses, zoonotic viruses that can hop from animals into humans. So anyway, it's a huge topic and a very interesting one. And I'm sure others will have something to say about it as well.
Great, thanks, Rob. Does anybody else want to add any context on the issue of modeling and pieces, missing parts of the modeling puzzle that might be critical and might be accessible?
Right. I'd like to talk a little bit about herd immunity, because that's what a lot of the models are based on. Once we sort of understand what the goal is and where we're working towards, a lot of the modeling begins to be understood, and we also begin to have some belief in what the models actually mean. What I've shown here on this slide is, on the very left, there's a virus infecting a population of hexagons. And as it infects them, they turn orange. And this is what's happening with CoV-2. This virus was originally doing quite comfortably among bats and replicating there. But once it got out of bats and into the human population, it's a new species, this virus turned rogue. And the reason it turned rogue was that none of us in the human species had seen this virus before, and we did not have the specific protection against it. And so this is what we're experiencing now, is the spread of this virus. And among the people infected, there will be some, thankfully not all, who are killed by the virus. As we progress through this interaction, we will of course have some people who have experienced the virus, and then they recover, and hopefully they have developed antibodies, those that Pamela talked about. And those are the striped individuals that you see here, the striped hexagons. Now this can go on for some time if it runs its natural course, and soon the population will get more and more people who are making antibodies and hopefully protected from the virus. That's when we call it that it's herd immunity, because you can see that having so many striped hexagons actually prevent the virus from reaching those who are uninfected, which are the empty hexagons. Now, we can change this dynamic here by pulling apart these triangles and separating the individuals from one another as far as possible, or isolating them, and that's what we've been doing. Now, we can also change this if we have drugs or vaccines. We could actually produce many more striped individuals. And so we need the numbers. We need to know what is going on in what we're experiencing and how it occurs within a population in any location. And since some of us are in very dense populations, we're very close together, the dynamics will be different, because the passage of the virus from one person to another will be faster because they're closer and easier for the virus to find. If we pull apart these triangles of individuals, then we have done what we have all experienced and are probably tired of, which is that we have physically distanced ourselves from each other. And of course, we are waiting for a vaccine that would also
help us so that more people will be protected. And then we can actually protect the very vulnerable among us and never have them exposed to the virus.
Thanks, Alice. That's very helpful and a good tutorial on herd immunity for the broader group of participants.
I think there's one thing that ought to be emphasized from that discussion, which is the critical role of testing. We need to know how many people in any given triangle are protected. And we can only know that by testing them for antibody. We need to know how many are actively infected and can pass the virus on to others. And we can only know that by testing for the virus itself. We should have, in the United States, a testing regime which gave a view of what the whole population looks like at any one time. And so we have to test not only today, but we have to test again next week and the week after and the week after to see whether we're controlling the spread of this virus. And we do not have the technical capability to run all of those tests today. What we need is to commit ourselves to getting the machines, the materials, and the analytic capability so that we can know all of those parameters and take advantage of what Alice just showed us.
Thanks. Agreed. Test, test, test. Test, test, test.
It's not going to be easy to do that because the numbers would be huge, and
Numbers are daunting.
Yeah.
right, they're daunting. In fact, I have another slide, which is the Rockefeller Foundation slide.
I'll share, yeah, maybe I'll share that one now. One moment.
I was on the board of the Rockefeller Foundation some years ago, so I get materials from them. And it turns out that they recognize that among the scientific community, there's been a consensus pretty much that we need testing, and a lot of it. And what they've done is to call together a large number of very good scientists to figure out how we can carry this out and what is really needed. And their take, which I think is really important, is that we don't have to make a decision between our health and our economy. That we really have three strategies that we have to keep in mind, and these are preventing death in people infected by COVID-2, maintaining our healthcare in the United States in such a way that they will be able to perform adequately, and not just adequately, but at their very best. And thirdly, we have to bring back the lifeline in our economy that sustains our lifestyle. And they are all important. And to get started on doing that, and the one way that we can do it that would be fair and reasonable is to think about testing and using the testing in the way that has been shown here. And the action plan is fairly straightforward, but the shocking thing is when you calculate the numbers about tests, we realize that we're testing somewhere in the thousands of tests a week. The group at Rockefeller Foundation figured that we really need to upgrade that to 1 million tests a week, and eventually make that up to 30 million tests a week. That's really going to be demanding and also needs coordination. With the tests, obviously the results have to be made public, and the database, that's number 3 here, and it's shared widely so that everyone can use it.
Mm-hmm.
The most important is that should there be a hotspot that develops anywhere,
Sure.
we can immediately send in a group to contain the spread of that virus. And as we know the numbers, we can make decisions as to how much to open and how much to allow people the freedom to move around. And also when we need to jump on it so that we can contain it.
Thanks. Okay, so we've got about 10 more minutes before, maybe a little less than 10 minutes, just to give all of you a heads up before we move on to the Q&A with the participants. So I want to preserve as much time as I can for that. But there's an issue that I want to connect. This is Earth Week, and we are thinking not just about coronavirus, but also about climate change and the environment and the intersection of the climate crisis and global health crises. So I'd like to start by directing this to Pamela. Pamela, if you'll be okay with that. So as I just said, the climate crisis is already a global health crisis. Some 7 million people are thought to die prematurely each year from poor air quality, and hundreds of millions more people now are hit by heat waves than 20 to 30 years ago, heat that can cause heat stroke in the frail, kidney damage, and congestive heart failure. If you connect the dots, as we've seen, these and other types of underlying conditions make the victims of COVID-19 highly susceptible to its most severe effects. And so there is a direct link between environmental air quality, the climate crisis, and at least how we experience the severity of an infection. But it could also be, and I think many have anticipated that with global warming and the uncertainties associated with it, that new types of pathogens, new types of viruses, bacteria, other pathogens will arise that we're going to have to be ready to deal with. So Pamela, what are some specific pathogen crises you worry about most, where global warming may or admittedly may not complicate our predictions and exacerbate their prevalence? And I'm also thinking, most people are worried about viruses that attack humans, but viruses attack plants. And with global warming, there could be very uncertain impacts on plants and in particular crops that can impact the global food supply chain. And so all of this is so interrelated. And I think I just would like to get your thoughts about what worries you the most and any thoughts you might want to share on this.
Okay, well, that's a very open-ended question.
Yes,
maybe you can pull up my slide. What I worry about are bats. So I'll just be specific here, is that coronaviruses, at least some of them, originate in bats. And as you may have heard, there's the slide. This is from the Washington Post about a week ago, called Climate Change Affects Everything, Even the Coronavirus. And so in the last 20 years, we've had 3 pandemics come out of coronaviruses that originated in bats, and that would be SARS, MERS, and now the virus is SARS-CoV-2 that's causing COVID-19. And so the problem, I think, is a very complicated one, which is that humans are interacting differently with animals all over the world, and animals serve as vectors for viruses. It's not just bats that pass viruses into the human population. Non-human primates do too, cattle do, rodents do, and it goes on. And so we're just interacting with the world and animals in the world very differently and crowding them out of their natural habitats. So as they move to different habitat, that gives viruses a chance to evolve. And one example would be, if you think about, if you've seen those pictures of the bats in the bat caves, I mean, they're just hanging there within inches of each other, absolutely no social distancing in that case. And they're just transmitting viruses back and forth between them. And they actually have a really incredible immune system, so that they're flying mammals, right? So they have to turn their metabolism down. So their immune system just kind of says, oh, okay, I have a virus. I don't really care. And so what may not be obvious here is that what's causing the problems in COVID-19 disease is our own immune system. That's what's creating this, if you've heard of the term cytokine storm and things like that, that lead to acute respiratory distress syndrome. It's your immune system that's doing this. Well, the bat just tolerates all these viruses and they mix between bats and they pass them to each other. And at some point they go on maybe to a secondary animal host, but sometimes they pass into humans. We've had 3 chances in 20 years. SARS and MERS kind of fizzled out. This one does not yet look like it's fizzling out. So what I worry about is climate change is going to move animals around. It's going to change vegetation, as you said, Jonas. It's going to affect what viruses infect plants. It's going to cause, viruses evolve really fast because they have a lot of progeny. And so they can just adapt much quicker than animals and humans can. So we're going to have new animal to animal interactions, new animal to human interactions. And then I'll just close by saying that clearly for a respiratory disease like COVID-19 or influenza for that matter, this is not good if you live in a place where your air quality is really poor. So that's going to be a terrible risk factor for people in the world who live in places where the air quality is bad anyway.
Thanks. It's helpful and also sobering to listen to. And
well,
we're
just
not.
let me say that I want to give a pitch here for basic research. I forgot.
Yeah, I was going to nudge you on that. So give us that.
So like, you know, I sit on National Institutes of Health, a study section to review grants. So what do you think would have happened a year ago if you put in a grant saying, I want to study the bat immune system? Nope, nobody would care. But you know what? That's the kind of basic research that we have to understand, because we're not going to understand what kinds of viruses are arising in these animals, how do they tolerate them, how likely are they to mutate so that they can move on to animal and then human hosts. And so there's a lot of basic research that has to be done. And I hate to see people thinking that, for example, in the non-scientific community, it may not sound like a good thing to be studying the bat immune system and bat viruses, but it is very good. You really need basic research into all kinds of hosts for viruses so that we can be better prepared, because it was already known if you sequence the viruses from bats, you can find 20 or more SARS-like viruses that could pass at any point into humans by a few mutations to switch which receptor they can bind to.
Okay, well, hopefully the NIH program managers are listening in, because I think everybody wants to learn about the bat immune system right now. At least I do. So thanks for that. I'll just,
can I
yeah,
jump
go
in?
ahead, Rob. Yeah.
A little tiny thought that maybe people will want to go read about, which is another very interesting animal system, and that has to do with the Rinderpest in Africa. Just for people, I think there's probably a lot of people here that actually are interested in the Serengeti.
Can you just spell that for those of us that haven't heard of that pest?
R-I-N-D-E-R, pest.
Thank you.
Rinder. And the thing I was going to say is the eradication of the Rinderpest led to a change in the number of wildebeest to the tune of around 800,000. I'm not going to say anything more, but just people can go look it up. The person's name to look up is Anthony Sinclair. But I think it's a very interesting example of the interplay between viruses and animals, and also just the general question of human impacts, because that was actually borne by cattle.
Great, thanks. And I'll just round it out because we don't have time to get into it. But as we talk about other pathogens, let's just keep it on everyone's minds that we haven't even gotten into mosquitoes and malaria and increased anticipation of that with climate change, the spread of ticks and the bacterial diseases that they spread that are also moving in ways that we don't yet fully understand or can't predict based on a warming climate. And so, you know, every cross section of life is impacted by climate change, and in particular when you think of climate change as a global health crisis. So I want to now ask Xenia Amashukeli to help facilitate some Q&A for all of you in this context.
Great, thank you, Jonas. Thank you, panel. We got a lot of questions, and a lot of them are very interesting, and we'll try to get to as many of them as we can, and I apologize in advance if we can answer all of them in the next 10 to 15 minutes. So I would like to start out with questions that focus mostly on development of the vaccine, and a lot of the participants have been asking questions about that. Specifically, based on what you currently know, what is the realistic timeline for the development? What kind of difficulties and problems that are typically what you can anticipate having the information that you currently have? And if this vaccine is developed at some point, do you think it's going to be one of those long-term applications, or is it something that people have to continuously work on, every year, annually, et cetera? So we can start there. If one of you would like to take the lead.
Great. Pamela, do you want to chime in to start on this topic?
Yeah. I'm actually optimistic that a vaccine could be developed, because the mutations don't bother me in SARS-CoV-2, because the mutation rate is about the same as in measles virus, and we have a very effective vaccine against measles. And actually, if you're infected with measles, you have lifelong immunity. So it's lower than flu, for which we have to have annual re-injections of a vaccine depending on what strains are circulating. That being said, there's a very complicated aspect of how vaccines work and what viruses do when they infect you, and that is induction of so-called immunological memory. So measles, for some unknown reason, induces really good immunological memory, so that you could see the virus 60 years later and then still be immune to it. There's some, from what I've read, coronaviruses don't tend to be that way. Common cold coronaviruses, we just keep getting over and over again, even if they're closely related strains. But keep in mind, we don't get that sick. So they're quite attenuated at this point. So I would hope that if we give a vaccine and it doesn't completely prevent COVID-19 disease, or it doesn't completely prevent you from, say, getting infected. And maybe it has to be administered every year, but I would hope that it would prevent this kind of really severe disease. And if we could do that, I think we'd be very happy. But as David said, can you imagine having to vaccinate even half of the world's population? Or maybe, referring to Alice's herd immunity, like 70% of the world's population? I mean, I don't know how that would be done in terms of manufacturing, let alone if you proved it was safe, you proved it worked, you proved it didn't have any harmful side effects. How would you do the manufacturing? That's a really interesting and difficult challenge.
Thank you. Does anybody else want to add anything to this answer? No? Great. Okay, so another group of questions focused on basically other environmental effects, and perhaps how that can affect the modeling and decision making. So you probably have heard about how temperature or humidity can affect possibly the potency or transmission rates, the survivability of this virus. So perhaps this is the question maybe for Rob or Alice. Do you know if any of that data is available and how it's being incorporated in the current models, or if there is a plan to incorporate that information into the models going forward?
I'll just quickly say something, which is, sorry to be self-referential, but a few weeks ago, we put out a paper called SARS-CoV-2 by the Numbers, which is available at the eLife website. And one of the classes of experiments that we talked about, they're not definitive because they are statements about the survival of virus on cardboard, on steel, on copper, in aerosols, and all that stuff. The references are there and people can look them up. But as with many of these things, it's actually a long chain of reasoning and experiment, from those experiments to knowing whether or not that's the way that you get the virus, and et cetera, like what's going to happen in the summer. So anyway, that's a useful reference point. And maybe Alice wants to amplify, or somebody else.
Yeah, there's been a long debate as to why we only get influenza viruses in the winter. Some people have said it has due to vitamin D. Other people say that it's due to the fact that people gather together, schools start in the fall, and you're getting a denser group of people, and therefore it begins to transmit from person to person. And so that's come down to the fact that a lot of people accept that just higher temperatures, warmer climates would prevent influenza. And it may well be, there are beginning to be studies on how fast droplets dry up and how long viruses stay at different temperatures. And there is one study that I ran across recently that was done at Children's Hospital in Philadelphia, in which they are modeling based on temperature. And what's interesting is when we look at the world distribution of cases of CoV-2, of people who get the virus, we find that much of the infection is in the Northern Hemisphere now. And we wonder what's going on in the Southern Hemisphere. And many of us think that maybe the testing has not been done down there as much as up here, and in the southern countries, they may not have had the wherewithals to do the testing. On the other hand, it could well be that as the weather changes down there, that the disease would be much worse and the spread would be much worse. I think these are all things that we need to learn about it, about the virus and its susceptibility to temperature and its insidious way of passing from one person to other persons, and whether weather affects that very much.
Thank you. So there's another group of questions that our participants would like to know. Based on, again, what you currently know, why there's so many people who show no symptoms when they are exposed to this virus and then test positive for COVID-19?
I think that goes into the category of questions we'd love to know the answer to, and don't. We don't have enough workup of individuals who clearly have been infected but had no symptoms. We don't understand why children seem to get infected but don't show symptoms. And we don't know whether the amount of virus they have and the way they have the virus enables them to transmit it to their parents and to other children, or whether in fact they are sort of dead ends. We don't understand why older people, we do understand to some extent why older people have more severe disease, but we don't understand that in detail. And the cytokine storm, which is such an important part of the viral disease process, is something that we're just beginning to work on. Not me, but people in general. So there's an enormous amount about the natural history of this virus that we don't know because we've never had experience with it before. It's never been in the human population. And so it's going to find its way in with a unique perspective no other virus is a model for this virus.
I just wanted to add, how would we know how often we're infected with influenza virus and we don't have symptoms? Like none of us would ever get tested. We're infected with all kinds of viruses, actually, all of us right now, and most of them aren't doing anything to us. So I don't see, we don't have a precedent for addressing this really.
When I grew up, the thing we worried about was poliovirus and getting the disease poliomyelitis. And it was very much like this situation because the bulk of people, particularly young children, who got infected with it and pass it on, had no symptoms. And so it was a transmission with no knowledge of somebody having been infected. And that's where we are today with this virus. But we have much better tools. We couldn't do the kind of testing in the 1950s, 1940s, when I was a child, that we can do today. And we ought to be taking advantage of the incredible technology that we have available to us today.
Virologists always have a name for an observation, especially when they don't really understand it. And so whenever I teach, there's always the iceberg phenomenon. That is, that you see the disease as the tip of the iceberg, but underneath there's just so much more going on and so many more people infected that we have no idea of what's happening.
Right.
Great, thank you. So I think we might have another couple of minutes for one more question, perhaps. So one of the questions, well, group of questions, I guess, if you were to do this way, talks about what is the best way to communicate a lot of the technical information, a lot of the detailed technical information about this particular virus and how it's affecting people to general public, and how to better communicate this message so it gets translated to the policymakers. And so if you have any ideas about how to go about it, it would be great if you can share those with us today.
I'll chip that one over to the panel. I'm going on mute.
I think David and Alice should answer that one.
Well, I mean, that's what we're doing here today. And this is something that scientists understand. They have responsibility to inform the public about what they know, what they don't know, what they need, what they don't need. And we're doing that continually. And particularly now that all of us are being held in our homes, we have nothing better to do than to try to communicate. And so there are lots of opportunities. Every scientific journal is opening itself to the public. You can read on almost a daily basis what's appearing in the New England Journal of Medicine and other journals if you want to know. And much of that is written in a way that's understandable. It really is a matter of our representatives, our government, taking cognizance of what's going on and committing itself to solving this problem. It's not an insoluble problem, but it takes organization at a much higher level than we have been willing to do.
Thanks, David. I think before we head to the break, rather than do one more question, there was one slide that Alice had prepared that I didn't show. And I do want to end with Alice's glass half full. It's great to have Alice on a panel to bring up, we're in a tough space right now and everybody's feeling it. And we try to be hopeful, but all of us can acknowledge our frustration then that it can be difficult to be hopeful. But I want to just show Alice's glass half full slide, which we didn't get to. Alice, why don't you just read off your, we don't even have to discuss them, just read them off and then we'll go to break so people can have something nice to think about during the break.
All right, I am a person that always sees my glass as being half full. So have all of you noticed how clean the air is in Pasadena? It's just beautiful out there and clear, and the flowers are blooming, and I think that this is one positive thing that we can all enjoy. On a more serious side, we are being forced to use more telemedicine because we don't want to go to the hospitals ourselves if possible. And this is a direction that is good for medicine. It will save time for everybody, and it might even save money. And then I don't think all of us are sitting around looking at Netflix all the time. Some are actually thinking of new things, and in business, there are some imaginative ways of selling food and wines. And I've been reading about virtual tastings. The American Society for Microbiology has suggested that we should establish a new National Guard in the future for medical and technical personnel so that we can meet surges such as what we're experiencing now. And teachers, all the professors at Caltech have been getting training in how to teach at a distance. And I've been noticing that even kindergarten teachers are trying very innovative things. And the South Carolina supply for teachers have come up with ideas of teaching laboratories at a distance, and I'm very interested in seeing how that turns out. Also, in the end, we have noticed that people are in collaborations. As Rob mentioned, there are just all sorts of people, physicists, thinking about droplets floating around in a closed room, and ideas of all kinds that are drawing people together. But I thought that you may have noticed also, which is most important to me, was that when we look at the essential personnel that's around, a majority are women, and many of the women are of color. And I hope that we will recognize that these people have been really important and essential in keeping us all going. And I hope that they will be paid for what they're really, really worth to all of us.
Thank you. With that, we're going to segue to our break. I want to thank the first set of panelists and remind everyone that the next panel is going to be equally enriching. And I'll leave it at that because I know people need a few minutes to take a break, but please come back and tune in. There's a lot of good content to come. Thanks again to all of our first panelists. That was terrific.